Dear students, today we are going to see oxytocin and vasopressin. It is coming under endocrine. So before going to see these two, first we have to know about pituitary gland. Already we have known this pituitary gland is otherwise called as hypophysis. It is having two parts. One is the anterior part, another one is the posterior part. This anterior part is otherwise called as adenohypophysis and posterior part is otherwise called as neurohypophysis. The hormones secreted by means of adenohypophysis are adrenocorticotropic hormone, thyroid stimulating hormone, follicle stimulating hormone, luteinizing hormone, prolactin and growth hormone. The hormone secreted by means of neurohypophysis, that is the posterior part of pituitary, are oxytocin, vasopressin, alpha MSH, that is melanocyte stimulating hormone, beta MSH and cocaine. Now we'll see the structure of the pituitary gland. So this was the structure of pituitary gland. So here this is the posterior part of pituitary, this is the anterior lobe of pituitary. This posterior part is formed from the posterior pituitary is formed from the nervous tissue and have nerve cells. These neurons have the cell bodies in the paraventricular nucleus and the supraoptic nuclei of the hypothalamus. Their axons, this would be the axons, these axons form a bundle and that bundle can be called as hypothalamo-hypophyseal tract. So from by means of this, here the hormones would be released from the posterior lobes and these hormones can be called as neurohypophyseal hormone or posterior pituitary hormones. They are oxytocin. Already we have seen that oxytocin, vasopressin, etc. So here this would be the anterior part of the pituitary and here by means of these two ways we can get the hormones. So these are the hormones secreted by means of anterior part of the pituitary. So now we are going to see oxytocin. So this oxytocin is a peptide hormone. So it is a combination of amino acids. It is a nonapeptide, that is it is having 9 amino acids. So what are the 9 amino acids? First one is cysteine, tyrosine, isoleucine, glutamine, asparagine, again cysteine, proline, leucine and finally glycine. So here first and sixth would be the same one, that is cysteine. Cysteine is a sulfur containing amino acid. So this sulfur is present in this cysteine and this sulfur is present in this cysteine. So because of the presence of this sulfur, they form the first and the sixth one, no, form a disulfide linkage. So because of the formation of disulfide linkage, it forms a ring. So here the first 6 amino acids form a ring that is closed by means of a disulfide linkage. Now we have the last 3 amino acids. No? So what are the last 3 amino acids? Proline, leucine and glycine. They form the tail portion. And this oxytocin is named because of its oxytocic action. So what are oxytocics? So these are the drugs which stimulates the smooth muscle of stress. Now we'll see the action of oxytocin. So during childbirth, the increased amount of oxytocin is released by the posterior pituitary gland into the bloodstream. So in response to the stretch receptors in the uterine cervix. So that causes uterine contractions that force the baby's head into the cervix. Then sensory impulses are generated. They travel to the control center in the hypothalamus and that is stimulating the posterior pituitary gland and they release more oxytocin. Again there may be uterine smooth muscle. So stronger contractions that force the baby's head further into the cervix. So in turn this stimulates more contractions and the greater stretching of the uterine cervix. And finally, inhibition occurs after delivery when uterine contractions no longer dilates the cervix. So now we'll see the SAR of oxytocin. Already we have known the structure of this oxytocin. It is having 9 amino acids. The first 6 form a ring and the last 3 form the tail portion. So here the amino acids 3, 4, 7, 8. So what are the amino acids? 3, 4, 7, 8. So these amino acids are not involved in hydrogen bonding. That's why they are determining the ring conformation. So they are acting as the binding sites for oxytocin receptors. So which are the amino acids that are acting as binding sites for oxytocin receptors? They are 3, 4, 7, 8. That is isoleucine, glutamine, proline and leucine. The second amino acid tyrosine that is having hydroxy group, and the fifth one that is called asparagine that is having amide function. So these two are essential for activity. Then the amino acids at 3, 4, 8, that is 3, 4, 8, and these three can be changed. That is, we can change the amino acids at 3, 4 and 8. If we are changing, we can get more selective compound. For example, in fourth position we have glutamino that can be replaced by means of threonine, or seventh one that is proline can be replaced by means of glycine, means we can get more potent oxytocic or antidiuretic active compound. So that's all about SAR of oxytocin. Now we'll see the uses of oxytocin. It is used in incomplete abortion after 20th week of gestation, used to prevent or control hemorrhage, and used to correct uterine hypotonicity. It is used to promote milk ejection, that is, it is acting by contracting the myoepithelium of mammary gland. It is administered parenterally by IV infusion or IV injection or IM injection. Oxytocin citrate buccal tablets are also available. Here, topical administration, that is like a nasal spray, that is also used to promote milk ejection, that is 2 to 3 minutes before nursing it can be used. So that's all about oxytocin. Now we are going to see about vasopressin. Vasopressin is an antidiuretic hormone. So what is the other name for vasopressin? It is antidiuretic hormone. There may be two types of vasopressin. One is arginine vasopressin, another one is lysine vasopressin. So it is structurally similar to oxytocin. Now we'll see the structure of oxytocin. Already we have seen that. So first it is having 6 amino acids that is forming a ring by means of disulfide linkage, and the tail portion is having 3 amino acids. So here how it is structurally similar to oxytocin. So this would be the structure of vasopressin. You compare these two. So here in third position oxytocin is having isoleucine. So that is replaced by means of phenylalanine. And eighth is leucine, no? So that leucine is replaced by means of arginine, means that is called as arginine vasopressin. And that leucine is replaced by means of lysine, means that is called as lysine vasopressin. That's all. Only two changes, one at the third, another at the eighth. In third, isoleucine is replaced by means of phenylalanine, and eighth leucine is replaced by means of arginine or lysine. If it is by means of arginine, that can be called as arginine vasopressin. By means of lysine, means that can be called as lysine vasopressin. And here that lysine is L-lysine, L-arginine. What is the physiological role of that vasopressin? That is regulation of water reabsorption in the renal tubules. In high doses, it promotes the contraction of arterioles and capillaries and increases the blood pressure. Now we'll see the analogues of vasopressin. 3 analogues are there. They are desmopressin, felipressin and lipressin. First we'll see desmopressin. It is having same structure as the top arginine vasopressin. In arginine vasopressin at 8th position we have L-arginine, but here we have D-arginine. It is a synthetic derivative. L-arginine is replaced by means of D-arginine. And one more changes. Usually we have seen first amino acid is cysteine, no? So that cysteine. In that cysteine is an amino acid. Usually amino acid means that is having two parts. One is amino group, another one is acid groups. This would be the structure of cysteine. So here that amino group of cysteine would be removed. That's also, instead of that we'll have one more H. So what is there? SH, CH2, CH2, COOH. So here SH. So that H is attached here. H is replaced, and that's why these two S, no, that would be connected. So S, CH2, CH2, CO. Here it is forming a bond here. So here it is not having one amino group. So that's why it is a 1 desamino AD-arginine vasopressin. So because of that deamination, it is resistant to the action of the enzyme called peptidase. Parent should have L-arginine, but here we are having D-arginine. No, that's why it is responsible for lower vasoconstrictive effects. Its efficacy, ease of administration, longer duration of action and lack of side effects makes the drug of the choice for the treatment of diabetes insipidus. It can be administered by IV or IM, used in the treatment of temporary polydipsia and polyuria associated with trauma. We'll see the next derivative that is called as felipressin. It is a lysine vasopressin. First one we have seen that is called arginine vasopressin, but this is a lysine vasopressin. One more change is in lysine vasopressin, second position should have tyrosine. No? So that tyrosine is replaced by means of phenylalanine. So it is the lysine vasopressin. Second tyrosine is replaced by means of phenylalanine. It is having low antidiuretic activity and a little oxytocic activity. It is 5 times more effective as a vasopressor than lysine vasopressin. It is used in surgery to minimize blood flow, mainly in obstetrics and gynecology. Now we'll see the last derivative that is called as the lipressin. It is a synthetic L-lysine vasopressin, that is having same structure as the top L-lysine, but it is a synthetic derivative. It is more stable and rapidly absorbed from the intestinal mucosa. It is available as a topical solution and spray. So that's all about oxytocin and vasopressin. Thank you.